Welcome to this explainer. Today, we're unpacking a truly groundbreaking 2026 Nature study. We're going to explore a radical new scientific framework that essentially treats the human immune system like a computer, one that is in desperate need of a software update. Let's get right into it.
So think about this massive biological mystery that has totally stumped scientists for years. When we face a chronic disease like cancer, why is it that some of our immune cells fight relentlessly while others just, shut down and give up? To figure this out, we really need to look closely at two seemingly identical cells that have completely opposite behaviors.
Here's our roadmap for this explainer. First, we'll look at the T-cell identity crisis. Second, finding the master switches. Third, unmasking the hidden regulators. Fourth, from theory to therapy. And finally, the future of immunotherapy.
All right, Section 1, the T-cell identity crisis. The sheer biological frustration these two cells have caused researchers is honestly immense. Just look at this. On one hand, we have terminally exhausted T cells, or TX term. These guys are completely dysfunctional and perform terribly against tumors. On the other hand, we have tissue-resident memory T cells, or TRM. Now, these are highly protective, driving incredibly strong anti-tumor immunity. But here is the massive catch. Despite having basically identical-looking biological profiles, they share very similar transcriptional and chromatin states, these two cells have entirely opposite functional outcomes on the battlefield. Historically, separating the fighters from the quitters has been nearly impossible.
Which brings us to Section 2, finding the master switches.
To finally tell these cells apart, researchers realized they couldn't just look at the surface level anymore. They had to dig deeper into the internal code driving their behavior. So they zeroed in on transcription factors. You can think of transcription factors as the biological master switches, or, the internal software engineers of the cell. They're the underlying regulators that literally dictate an immune cell's differentiation state. They ultimately decide its true identity and whether it's going to fight or just surrender.
Now, here is the absolute crucial point of this study. Just because a transcription factor is expressed doesn't mean it's actually functioning. Think about it like this. Just because an employee is sitting at their desk doesn't mean they're the CEO making the big decisions, right? Standard differential expression testing just looks at who is physically in the building. But the researchers needed a way to figure out who is actually calling the shots and giving the orders.
To find the actual CEOs in charge of these cells, the research team built this incredible multi-omics framework called TIG. They mapped out an unusually strong progression here. They started by building a massive atlas, then ran their TIG algorithm to infer the real driving transcription factors. And finally, they actually physically validated those predictions using in vivo CRISPR screening.
And that number right there brilliantly illustrates the massive scale of their very first step. They didn't just look at a handful of cells. No, they integrated RNA and ATAC sequencing across 121 different samples, mapped across 9 distinct T-cell states. It's like they built a towering mountain of data just to make sure their algorithm had every single thing it needed to spot those hidden patterns.
Moving on to Section 3, unmasking the hidden regulators.
By using the PageRank-based TIG algorithm as a sort of computational magnifying glass, the researchers moved way beyond simple expression. They looked at the entire regulatory network structure to unmask the hidden culprits that were driving cell exhaustion. They basically asked, what transcription factor changes exhaustion while preserving the good, beneficial memory? And they categorized these newly discovered switches. What's wild is they revealed that ZSCAN20 and JDP2 are primary drivers of exhaustion. This is huge because neither of these had any previously known role in T cells, zero. They also flagged KLF6 as a protective TRM selective regulator and found that HIC1 and GFI1 act as shared regulators, playing really complex roles in both states.
Okay, Section 4, from theory to therapy.
It is one thing to have a neat computational prediction, but it's a whole other ballgame to actually test it in a living organism. So the researchers used CRISPR to physically knock out those specific bad switches, ZSCAN20 and JDP2, in living mouse models. The results? A complete cascading wave of benefits. They successfully reduced the exhausted T-cell phenotypes while perfectly preserving those protective memory formations. The cells showed improved cytokine production, much better tumor control, and get this, they even synergized beautifully with existing anti-PD-1 therapies. They proved they could actually fix the exhaustion bug without breaking the protective memory programs.
Why does this matter to you and me?
Well, because these computational predictions weren't just some quirky phenomenon only found in mice. The authors went on to demonstrate that this exact transcription factor biology was conserved across human tumors too, successfully improving human T-cell function. That translates to incredibly compelling potential for real-world clinical applications.
But of course, as an objective explainer, it's really important we look at the full picture and note the limitations the authors themselves acknowledged. First off, the transcription factor classification here is probabilistic. There's no absolute threshold, so some misclassification is definitely possible. Plus, even with the human validation, the study still heavily relies on mouse models for the really deep mechanistic work. And crucially, the functional endpoints they measured were short to medium term. We absolutely still need to figure out the long-term persistence, safety, and any autoimmune risks associated with editing these cells.
Finally, Section 5, the future of immunotherapy.
As we reach the end of our roadmap, we have to look at the immediate medical horizons that the Taiji framework has just blown wide open. The authors envision a future where we use these exact findings to build combinatorial recipes. Imagine editing an immune cell by actively deleting the exhaustion drivers like ZScan20, while simultaneously overexpressing the beneficial memory factors like KLF6. The ultimate goal is to translate these exact recipes into advanced CAR-T and TIL therapies, essentially engineering the ultimate, exhaustion-resistant immune cells, all while expanding this incredible atlas to entirely new cell types.
I want to leave you with a provocative thought. We are actively moving from single-gene biology into full-blown regulatory architecture. If mastering the master switches of our cells allows us to essentially rewrite the software of the human immune system, what disease will we program it to defeat next? Thank you for joining me for this explainer, and keep questioning the science that shapes our future.